Maggot therapy, also known as larval therapy, is a type of biotherapy involving the introduction of live, disinfected maggots fly larvae into non-healing skin and soft tissue wounds of a human or other animal for the purpose of cleaning out the necrotic, dead tissue within a wound, debridement, and disinfection. There is evidence that maggot therapy may help with wound healing. 1. 2. Maggots in medical packaging. Maggot therapy improves healing in chronic ulcers. 1. In diabetic foot ulcers there is tentative evidence of benefit. 3. A Cochrane review of methods for the debridement of venous leg ulcers found maggot therapy to be broadly as effective as most other methods, but the study also noted that the quality of data was poor. 4. In 2003, the United States Food and Drug Administration (FDA) cleared maggots from common green bottle fly for use as a medical device in the U.S. for the purpose of treatment of 5. Non-healing necrotic skin and soft tissue wounds. Pressure ulcers. Venous stasis ulcers. Maggot therapy. Maggot debridement therapy on a wound from a diabetic foot. Other names: maggot debridement therapy, MDT, larval therapy, larva therapy, biodebridement, biosurgery. Edit on Wikidata. Medical uses. Neuropathic foot ulcers. Non-healing traumatic or post-surgical wounds. Limitations. The wound must be of a type that can benefit from the application of maggot therapy. A moist, exudating wound with sufficient oxygen supply is a prerequisite. Not all wound types are suitable. Wounds which are dry, or open wounds of body cavities do not provide a good environment for maggots to feed. In some cases it may be possible to make a dry wound suitable for larval therapy by moistening it with saline soaks. 6. Patients and doctors may find maggots distasteful, although studies have shown that this does not cause patients to refuse the offer of maggot therapy. 7. Maggots can be enclosed in opaque polymer bags to hide them from sight. Dressings must be designed to prevent any maggots from escaping, while allowing air to get to the maggots. 8. Dressings are also designed to minimize the uncomfortable tickling sensation that the maggots often cause. 9. Mechanisms of action. The maggots have 4 principal actions. Debridement 10. Disinfection of the wound 11. Stimulation of healing 11. Biofilm inhibition and eradication 12. Debridement. In maggot therapy, large numbers of small maggots consume necrotic tissue far more precisely than is possible in a normal surgical operation, and can debride a wound in a day or two. The area of a wound's surface is typically increased with the use of maggots due to the undebrided surface not revealing the actual underlying size of the wound. They derive nutrients through a process known as extracorporeal digestion, by secreting a broad spectrum of proteolytic enzymes 13 that liquefy necrotic tissue, and absorb the semi-liquid result within a few days. In an optimum wound environment maggots molt twice, increasing in length from about 2 mm to about 10 mm, and in girth, within a period of 48 to 72 hours by ingesting necrotic tissue, leaving a clean wound free of necrotic tissue when they are removed. 14. Disinfection. Secretions from maggots believed to have broad-spectrum antimicrobial activity include allantoin, urea, phenylacetic acid, phenylacetaldehyd, calcium carbonate, proteolytic enzymes, and many others. 15. In vitro studies have shown that maggots inhibit and destroy a wide range of pathogenic bacteria including methicillin-resistant Staphylococcus aureus. Disinfection. Secretions from maggots believed to have broad-spectrum antimicrobial activity include allantoin, urea, phenylacetic acid, phenylacetaldehyd, calcium carbonate, proteolytic enzymes, and many others. 15. In vitro studies have shown that maggots inhibit and destroy a wide range of pathogenic bacteria including methicillin-resistant Staphylococcus aureus, MRSA, group A and B streptococci, and gram-positive aerobic and anaerobic strains. 16. Other bacteria like Pseudomonas aeruginosa, E. coli or Proteus spp. are not attacked by maggots, and in case of Pseudomonas even the maggots are in danger. 17. Biology of maggots. Lucilia serracata, green bottle fly. Protoformia terraenae, northern blowfly. Those flies whose larvae feed on dead animals will sometimes lay their eggs on the dead parts, necrotic or gangrenous tissue, of living animals. The infestation by maggots of live animals is called myiasis. Some maggots will feed only on dead tissue, some only on live tissue, and some on live or dead tissue. The flies used most often for the purpose of maggot therapy are blowflies of the Calliphoridae. The blowfly species used most commonly is Lucilia serracata, the common green bottle fly. Another important species, Protoformia terraenae, is also notable for its feeding secretions, which combat infection by Streptococcus pyogenes and S. pneumoniae. 18. History. Written records have documented that maggots have been used since antiquity as a wound treatment. 19. There are reports of the use of maggots for wound healing by Maya, Native Americans, and Aboriginal tribes in Australia. Maggot treatment was reported in Renaissance times. Military physicians observed that soldiers whose wounds had become colonized with maggots experienced significantly less morbidity and mortality than soldiers whose wounds had not become colonized. These physicians included Napoleon's general surgeon, Baron Dominique Larrey. Larrey reported during the French campaign in Egypt and Syria, 1798-1801, that certain species of fly consumed only dead tissue and helped wounds to heal. 18. Joseph Jones, a ranking Confederate medical officer during the American Civil War, stated, "I have frequently seen neglected wounds filled with maggots. As far as my experience extends, these worms eat only dead tissues, and do not injure specifically the well parts." The first documented therapeutic use of maggots in the United States is credited to 2nd Confederate medical officer Dr. J. F. Zacharias, who reported during the American Civil War that Maggots in a single day would clean a wound much better than any agents we had at our command. I am sure I saved many lives by their use. He recorded a high survival rate in patients he treated with maggots. 20. During World War I, orthopedic surgeon William S. Baer recorded the case of a soldier left for several days on the battlefield who had sustained compound fractures of the femur and large flesh wounds. The soldier arrived at the hospital with maggots infesting his wounds but had no fever or other signs of infection and survived his injuries, which would normally have been fatal. After the war, Baer began using maggot therapy at Boston Children's Hospital in Massachusetts. 21-22, 169-71. There were reports that American prisoners of war of the Japanese in World War II resorted to maggot therapy to treat severe wounds. 23-24. A survey of U.S. Army doctors published in 2013 found that 10% of them had used maggot therapy. 25. Regulation. In January 2004, the FDA granted permission to produce and market maggots for use in humans or animals as a prescription-only medical device for the following indications: for debriding non-healing necrotic skin and soft tissue wounds, including pressure ulcers, venous stasis ulcers, neuropathic foot ulcers, and non-healing traumatic or post-surgical wounds. 26-27. Veterinary use. The use of maggots to clean dead tissue from animal wounds is part of folk medicine in many parts of the world. 28. It is particularly helpful with chronic osteomyelitis, chronic ulcers, and other pus-producing infections that are frequently caused by chafing due to work equipment. Maggot therapy for horses in the United States was reintroduced after a study published in 2003 by veterinarian Dr. Scott Morrison. This therapy is used in horses for conditions such as osteomyelitis secondary to laminitis, subsolar abscesses leading to osteomyelitis, post-surgical treatment of street nail procedure for puncture wounds infecting the navicular bursa, kinkor, non-healing ulcers on the frog, and post-surgical site cleaning for keratoma removal. 29. However, there have not been many case studies done with maggot debridement therapy on animals, and as such it can be difficult to accurately assess how successful it is. 30. References. 1. Sun, Xinhuan, Zhang, Kechun, Chen, Jinggan, et al. Model organism. A model organism is a non-human species that is extensively studied to understand particular biological phenomena, with the expectation that discoveries made in the model organism will provide insight into the workings of other organisms. 1-2 Model organisms are widely used to research human disease when human experimentation would be unfeasible or unethical. 3. This strategy is made possible by the common descent of all living organisms, and the conservation of metabolic and developmental pathways and genetic material over the course of evolution. 4. Escherichia coli is a gram-negative prokaryotic model organism. Drosophila melanogaster, one of the most famous subjects for genetics experiments. Saccharomyces cerevisiae, one of the most intensively studied eukaryotic model organisms in molecular and cell biology. Research using animal models has been central to most of the achievements of modern medicine. 5, 6, 7. It has contributed most of the basic knowledge in fields such as human physiology and biochemistry, and has played significant roles in fields such as neuroscience and infectious disease. 8, 9. The results have included the near eradication of polio and the development of organ transplantation, and have benefited both humans and animals. Saccharomyces cerevisiae, one of the most intensively studied eukaryotic model organisms in molecular and cell biology. Research using animal models has been central to most of the achievements of modern medicine. 5, 6, 7. It has contributed most of the basic knowledge in fields such as human physiology and biochemistry, and has played significant roles in fields such as neuroscience and infectious disease. 8, 9. The results have included the near eradication of polio and the development of organ transplantation, and have benefited both humans and animals. 5, 10. From 1910 to 1927, Thomas Hunt Morgan's work with the fruit fly Drosophila melanogaster identified chromosomes as the vector of inheritance for genes. 11, 12. And Eric Kandel wrote that Morgan's discoveries helped transform biology into an experimental science. 13. Research in model organisms led to further medical advances, such as the production of the diphtheria antitoxin 14, 15, and the 1922 discovery of insulin 16, and its use in treating diabetes, which had previously meant death. 17. Modern general anesthetics such as halothane were also developed through studies on model organisms, and are necessary for modern, complex surgical operations. 18. Other 20th century medical advances and treatments that relied on research performed in animals include organ transplant techniques, 19, 20, 21, 22, the heart-lung machine, 23, antibiotics, 24, 25, 26, and the whooping cough vaccine. 27. In researching human disease, model organisms allow for better understanding the disease process without the added risk of harming an actual human. The species of the model organism is usually chosen so that it reacts to disease or its treatment in a way that resembles human physiology, even though care must be taken when generalizing from one organism to another. 28. However, many drugs, treatments and cures for human diseases are developed in part with the guidance of animal models. 29. 30. Treatments for animal diseases have also been developed, including for rabies, 31, anthrax, 31, glanders, 31, feline immunodeficiency virus, FIV, 32, tuberculosis, 31, Texas cattle fever, 31, classical swine fever, hog cholera, 31, heartworm, and other parasitic infections. 33. Animal experimentation continues to be required for biomedical research. 34. And is used with the aim of solving medical problems such as Alzheimer's disease. 35. AIDS. 36. Multiple sclerosis. 37. Spinal cord injury. Many headaches. 38. And other conditions in which there is no useful in vitro model system available. Model organisms are drawn from all three domains of life, as well as viruses. One of the first model systems for molecular biology was the bacterium Escherichia coli, E. coli, a common constituent of the human digestive system. The mouse, Mus musculus, has been used extensively as a model organism and is associated with many important biological discoveries of the 20th and 21st centuries. 39. Other examples include baker's yeast, Saccharomyces cerevisiae, the T4 phage virus, the fruit fly Drosophila melanogaster, the flowering plant Arabidopsis thaliana, and guinea pigs Cavia porcellus. Several of the bacterial viruses bacteriophage that infect E. coli also have been very useful for the study of gene structure and gene regulation, e.g. phages lambda and T4. 40. Disease models are divided into three categories. Homologous animals have the same causes, symptoms and treatment options as would humans who have the same disease. Isomorphic animals share the same symptoms and treatments. And predictive models are similar to a particular human disease in only a couple of aspects, but are useful in isolating and making predictions about mechanisms of a set of disease features. 41. History. The use of animals in research dates back to ancient Greece, with Aristotle 384–322 BCE, and Erasistratus 304–258 BCE, among the first to perform experiments on living animals. 42. Discoveries in the 18th and 19th centuries included Antoine Lavoisier's use of a guinea pig in a calorimeter to prove that respiration was a form of combustion, and Louis Pasteur's demonstration of the germ theory of disease in the 1880s using anthrax in sheep. 43. Research using animal models has been central to most of the achievements of modern medicine. 5, 6, 7. It has contributed most of the basic knowledge in fields such as human physiology and biochemistry, and has played significant roles in fields such as neuroscience and infectious disease. 8, 9. For example, the results have included the near eradication of polio and the development of organ transplantation, and have benefited both humans and animals. 5, 10. From 1910 to 1927, Thomas Hunt Morgan's work with the fruit fly Drosophila melanogaster identified chromosomes as the vector of inheritance for genes. 11, 12. Drosophila became one of the first, and for some time the most widely used, model organisms. 44. And Eric Kandel wrote that Morgan's discoveries helped transform biology into an experimental science. 13. D. melanogaster remains one of the most widely used eukaryotic model organisms. During the same time period, studies on mouse genetics in the laboratory of William Ernest Castle in collaboration with Abbie Lathrop led to generation of the DB8 dilute, brown and non-agouti, inbred mouse strain and the systematic generation of other inbred strains. 45, 46. The mouse has since been used extensively as a model organism and is associated with many important biological discoveries of the 20th and 21st centuries. 39. In the late 19th century, Emil von Behring isolated the diphtheria toxin and demonstrated its effects in guinea pigs. He went on to develop an antitoxin against diphtheria in animals and then in humans, which resulted in the modern methods of immunization and largely ended diphtheria as a threatening disease. 14. The diphtheria antitoxin is famously commemorated in the Iditarod race, which is modeled after the delivery of antitoxin in the 1925 serum run to Nome. The success of animal studies in producing the diphtheria antitoxin has also been attributed as a cause for the decline of the early 20th century opposition to animal research in the United States. 15. Subsequent research in model organisms led to further medical advances, such as Frederick Banning's research in dogs, which determined that the isolates of pancreatic secretion could be used to treat dogs with diabetes. This led to the 1922 discovery of insulin, with John McLeod, 16, and its use in treating diabetes, which had previously meant death. 17. John Cade's research in guinea pigs discovered the anticonvulsant properties of lithium salts. 47. Which revolutionized the treatment of bipolar disorder, replacing the previous treatments of lobotomy or electroconvulsive therapy. Modern general anesthetics, such as halothane and related compounds, were 17. John Cade's research in guinea pigs discovered the anticonvulsant properties of lithium salts. 47. Which revolutionized the treatment of bipolar disorder, replacing the previous treatments of lobotomy or electroconvulsive therapy. Modern general anesthetics, such as halothane and related compounds, were also developed through studies on model organisms, and are necessary for modern, complex surgical operations. 1848. In the 1940s, Jonas Salk used rhesus monkey studies to isolate the most virulent forms of the polio virus. 49. Which led to his creation of a polio vaccine. The vaccine, which was made publicly available in 1955, reduced the incidence of polio 15-fold in the United States over the following 5 years. 50. Albert Sabin improved the vaccine by passing the polio virus through animal hosts, including monkeys. The Sabin vaccine was produced for mass consumption in 1963, and had virtually eradicated polio in the United States by 1965. 51. It has been estimated that developing and producing the vaccines required the use of 100,000 rhesus monkeys, with 65 doses of vaccine produced from each monkey. Sabin wrote in 1992, without the use of animals and human beings, it would have been impossible to acquire the important knowledge needed to prevent much suffering and premature death not only among humans, but also among animals. 52. Other 20th century medical advances and treatments that relied on research performed in animals include organ transplant techniques, 19, 20, 21, 22, the heart-lung machine, 23, antibiotics, 24, 25, 26, and the whooping cough vaccine. 27 treatments for animal diseases have also been developed, including for rabies, 31 anthrax, 31 glanders, 31 feline immunodeficiency virus, FIV, 32 tuberculosis, 31 Texas cattle fever, 31 classical swine fever, hog cholera, 31 heartworm, and other parasitic infections. 33 animal experimentation continues to be required for biomedical research. 34 and is used with the aim of solving medical problems such as Alzheimer's disease. 35 AIDS, 36 53 54 multiple sclerosis, 37 spinal cord injury, many headaches, 38 and other conditions in which there is no useful in vitro model system available. Selection. Models are those organisms with a wealth of biological data that make them attractive to study as examples for other species and/or natural phenomena that are more difficult to study directly. Continual research on these organisms focuses on a wide variety of experimental techniques and goals from many different levels of biology, from ecology, behavior and biomechanics, down to the tiny functional scale of individual tissues, organelles and proteins. Inquiries about the DNA of organisms are classed as genetic models with short generation times, such as the fruit fly and nematode worm, experimental models, and genomic parsimony models, investigating pivotal position in the evolutionary tree. 55 Historically, model organisms include a handful of species with extensive genomic research data, such as the NIH model organisms. 56. Often, model organisms are chosen on the basis that they are amenable to experimental manipulation. This usually will include characteristics such as short life cycle, techniques for genetic manipulation, inbred strains, stem cell lines, and methods of transformation, and non-specialist living requirements. Sometimes, the genome arrangement facilitates the sequencing of the model organism's genome, for example, by being very compact or having a low proportion of junk DNA, e.g. yeast, or Arabidopsis, or pufferfish. 57. When researchers look for an organism to use in their studies, they look for several traits. Among these are size, generation time, accessibility, manipulation, genetics, conservation of mechanisms, and potential economic benefit. As comparative molecular biology has become more common, some researchers have sought model organisms from a wider assortment of lineages on the tree of life. Phylogeny and genetic relatedness. The primary reason for the use of model organisms in research is the evolutionary principle that all organisms share some degree of relatedness and genetic similarity due to common ancestry. The study of taxonomic human relatives, then, can provide a great deal of information about mechanism and disease within the human body that can be useful in medicine. Various phylogenetic trees for vertebrates have been constructed using comparative proteomics, genetics, genomics as well as the geochemical and fossil record. 58 These estimations tell us that humans and chimpanzees last shared a common ancestor about 6 million years ago, Maya. As our closest relatives, chimpanzees have a lot of potential to tell us about mechanisms of disease and what genes may be responsible for human intelligence. However, chimpanzees are rarely used in research and are protected from highly invasive procedures. Rodents are the most common animal models. Phylogenetic trees estimate that humans and rodents last shared a common ancestor approximately 80 to 100 Maya. 59, 60 Despite this distant split, humans and rodents have far more similarities than they do differences. This is due to the relative stability of large portions of the genome, making the use of vertebrate animals particularly productive. Genomic data is used to make close comparisons between species and determine relatedness. Humans share about 99% of their genome with chimpanzees, 61, 62, 98.7% with bonobos, 63, and over 90% with the mouse. 60. With so much of the genome conserved across species, it is relatively impressive that the differences between humans and mice can be accounted for in approximately 6,000 genes, of approximately 30,000 total. Scientists have been able to take advantage of these similarities in generating experimental and predictive models of human disease. Use. There are many model organisms. One of the first model systems for molecular biology was the bacterium Escherichia coli, a common constituent of the human digestive system. Several of the bacterial viruses, bacteriophage, that infect E. coli also have been very useful for the study of gene structure and gene regulation, e.g. phages lambda and T4. However, it is debated whether bacteriophages should be classified as organisms, because they lack metabolism and depend on functions of the host cells for propagation. 64. In eukaryotes, several yeasts, particularly Saccharomyces cerevisiae, Baker's, or budding yeast, have been widely used in genetics and cell biology, largely because they are quick and easy to grow. The cell cycle in a simple yeast is very similar to the cell cycle in humans and is regulated by homologous proteins. The fruit fly Drosophila melanogaster is studied, again, because it is easy to grow for an animal, has various visible congenital traits and has a polytene giant chromosome in its salivary glands that can be examined under a light microscope. The roundworm Caenorhabditis elegans is studied because it has very defined development patterns involving fixed numbers of cells, and it can be rapidly assayed for abnormalities. 65. Disease models. Animal models serving in research may have an existing, inbred or induced disease or injury that is similar to a human condition. These test conditions are often termed as animal models of disease. The use of animal models allows researchers to investigate disease states in ways which would be inaccessible in a human patient, performing procedures on the non-human animal that imply a level of harm that would not be considered ethical to inflict on a human.